Chapter 12. Membrane composition determines resolution potential. Resolution potential is not created at the moment inflammation needs to end. It is built earlier at the level of membrane composition. That is the central claim of this chapter, and it changes the way the entire inflammatory sequence has to be understood. A tissue does not wait until it is inflamed and then decide from nothing whether it will escalate cleanly and resolve well. It enters the inflammatory event carrying a preloaded structural condition. Its membranes already contain a certain balance of saturated, monounsaturated, omega-6 and omega-3 fatty acids. They already possess a certain degree of oxidative stability or oxidative fragility. They already hold a certain ratio of initiation-ready substrate to resolution-ready substrate. They already provide a certain physical environment for receptors, transporters, channels, phospholipids, cyclooxygenases, lipoxygenases, and the broader machinery of inflammatory signaling. That means a cell's capacity to resolve is partly determined before the first neutrophil arrives. The membrane is not simply where inflammatory substrate is stored. It is where inflammatory outcome is biased in advance. This is why membrane composition has to be treated as more than a structural footnote. A membrane determines not only whether a cell can maintain gradients, host receptors, and preserve compartment boundaries. It also determines what kind of inflammatory program the cell can run under stress. A phospholipid pool enriched in arachidonic acid-heavy omega-6 derived substrate creates one kind of future. It favors early-phase eicosanoid production, stronger recruitment logic, and greater readiness for inflammatory amplification. A membrane meaningfully enriched in EPA and DHA creates another kind of future. It still permits initiation because the body still requires initiation, but it carries more of the precursor pool needed for mediator class switching and downstream generation of resolvins, protectins, and maresins once the tissue must stop escalating and start completing. The body therefore does not resolve from abstract nutrition. It resolves from the material built into its membranes. The phrase resolution potential is important because it emphasizes possibility rather than guarantee. A membrane rich in omega-3 derived substrate does not automatically produce strong resolution if the tissue is severely oxidized, chronically re-triggered, energetically depleted, or trapped in a cytokine environment that never allows phase transition. But without the right membrane composition, even the possibility of strong resolution becomes weaker from the start. The tissue cannot generate what it did not store. It cannot route into later-phase lipid mediator families if the releasable pool is too skewed toward early-phase substrate. It cannot preserve signaling precision during inflammatory stress if the membrane itself is chemically fragile and structurally unstable. Resolution potential, therefore, depends on both what the membrane contains and how well that membrane can hold together while the inflammatory event unfolds. Composition and stability are inseparable here. This also explains why highly unsaturated fatty acids occupy such a complex position in the story. EPA and DHA are essential to resolution potential because they provide the raw material for specialized pro-resolving mediators and alter membrane behavior in ways that can support cleaner phase transition. But they are also chemically vulnerable because their multiple double bonds make them easier targets for peroxidation. That means membrane composition determines resolution potential in two directions at once. A membrane too poor in omega-3 derived substrate is under-equipped for strong SBM generation. A membrane rich in omega-3 derived substrate but poorly protected from oxidative damage may carry theoretical resolution potential that is repeatedly degraded before it can be expressed. The body therefore needs more than omega-3 intake. It needs a membrane environment stable enough to preserve that intake as usable future biology. The relationship with omega-6 becomes clearer inside this framework. Omega-6 is not wrong. Arachidonic acid and related substrate remain necessary because inflammation has to be able to start. But a membrane overwhelmingly dominated by omega-6 derived releasable substrate creates a biased inflammatory future. It shifts enzymatic competition, increases the relative ease of eicosanoid-heavy initiation, and makes it harder for omega-3 derived completion pathways to gain sufficient downstream influence. In that sense, membrane composition does not merely affect inflammatory intensity. It affects inflammatory direction. It determines whether the tissue is easier to push into escalation than into closure. This is why modern diet matters so much. It is not just adding inflammatory foods. It is remodeling the membranes from which future inflammatory decisions will be made. Membrane composition also influences resolution potential through receptor behavior and signal interpretation. Inflammation is not driven only by what lipid mediators are released later. It is shaped from the beginning by how cells read the environment through membrane-bound receptors, channels, and signaling assemblies. A membrane that is too disordered, too oxidized, or too compositionally skewed can amplify danger signals more easily and normalize them more poorly. Receptors cluster differently. Microdomains behave differently. Transporters, phospholipids, and signaling proteins encounter different local conditions. The cell therefore interprets the same inflammatory cue through a different surface. This matters for resolution because a tissue that overreads danger at the receptor level will often enter more forcefully into initiation and may remain harder to shift out of it later. Membrane composition is not only setting substrate supply, it is shaping the behavior of the interpretive surface itself. The mitochondrial dimension matters here as well. Resolution is metabolically expensive, and mitochondrial membranes are highly specialized lipid structures whose integrity determines whether repair can be paid for. A tissue with unstable or damaged mitochondrial membranes may still initiate inflammation because initiation can be driven by acute signaling and stored substrate release, but it will struggle when the time comes to fund cleanup, efferocytosis, membrane repair, barrier restoration, and tissue rebuilding. In that sense, membrane composition determines resolution potential not only through inflammatory mediator biology, but through energy biology. A cell resolves from the membranes it stores and from the membranes it uses to generate ATP. If either level is structurally compromised, the later phase becomes harder to execute. This chapter also helps explain why chronic illness so often looks like repeated failure rather than a single overwhelming trigger. The membrane is the place where tissue history is stored. Long-term diet, oxidative stress, inflammatory repetition, lipoprotein traffic, hormonal state, mitochondrial strain, and repair capacity all leave their mark on membrane composition over time. The body therefore accumulates a structural inflammatory memory. A tissue repeatedly remodeled under omega-6 dominance, oxidative fragility, poor omega-3 incorporation, and failed cleanup becomes a tissue with lower resolution potential before the next insult even occurs. It is easier to activate, harder to protect, and less equipped to finish. That is why inflammatory chronicity often feels disproportionate to the present moment. The tissue is not responding only from present conditions. It is responding from a membrane history that has already lowered the probability of successful closure. Seen clearly, membrane composition determines resolution potential because the membrane decides what inflammatory future is physically possible. It stores the substrate reservoir. It shapes receptor behavior. It controls enzymatic access. It influences oxidative stability. It affects mitochondrial competence. It biases whether the tissue will be better at initiation, better at completion, or unstable in both directions. The body does not begin resolution from nowhere. It begins from the surface it already built. If that surface is rich in the right precursor material, sufficiently protected, and integrated into a broader environment capable of phase transition, then specialized pro-resolving biology can become more than theory. If it is unstable, omega-6 dominant, oxidatively damaged, and metabolically underpowered, then resolution potential remains low no matter how much the tissue might need it. That is the deeper message of this chapter. The ability to end inflammation is not primarily a matter of willpower, not primarily a matter of one acute intervention, and not primarily a matter of suppressing the first half harder. It is a matter of what kind of membrane the body has built over time. The tissue resolves from its architecture. If the architecture has been built for escalation, then the body will escalate more easily than it finishes. If the architecture has been rebuilt to support substrate balance, structural stability, downstream conversion, and energetic sufficiency, then the body gains something much more valuable than lower inflammation. It gains the capacity to complete inflammation as a successful biological program. EPA/DHA incorporation into phospholipids. EPA and DHA only become biologically decisive when they move from intake into structure. That transition happens through incorporation into phospholipids. Until then, omega-3 remains nutritionally present but physiologically shallow. Once these fatty acids are esterified into membrane phospholipids, they stop behaving like transient dietary inputs and start behaving like part of the tissue itself. They enter the bilayer, alter the physical environment surrounding receptors and enzymes, change what kinds of fatty acids can later be released during inflammatory activation, and create the precursor pool from which specialized pro-resolving mediators may eventually be derived. This is why incorporation matters more than consumption in the abstract. The body does not resolve from what was swallowed. It resolves from what was built into membranes and held there long enough to influence future signaling. This incorporation is not uniform across all tissues, and that matters because phospholipid remodeling is one of the ways the body assigns different structural priorities to different organs. DHA becomes especially concentrated in neural and retinal membranes, where its physical properties contribute to membrane curvature behavior, excitability, vesicle dynamics, and signal fidelity. EPA tends to be discussed more heavily in relation to inflammatory signaling pools and systemic mediator balance, though it also participates structurally once incorporated. The point is that these fatty acids are not simply floating in circulation performing benefits from the bloodstream. They have to be placed into phospholipid architecture in the tissues where their later structural and signaling roles will matter. A membrane enriched with EPA and DHA is not merely chemically different. Is carrying a different future response profile. That future profile changes because phospholipids are the storage format from which later inflammatory and pro-resolving language is generated. When phospholipase activity is triggered by tissue stress, receptor activation, microbial signals, oxidative pressure, or mechanical injury, the body releases fatty acids from the membrane positions it previously loaded. If EPA and DHA are underrepresented in those phospholipid pools, then the tissue has less omega-3 derived substrate available for downstream generation of resolvins, protectins, and maresins. If EPA and DHA are meaningfully incorporated, the tissue carries a larger reserve of completion-capable substrate. This is why incorporation into phospholipids is such a critical step. It changes not only membrane behavior in the resting state, but the biochemical options available once the membrane is mobilized during inflammation. The future mediator field is partly written into the phospholipid pool before the inflammatory event ever begins. Incorporation also changes membrane behavior before any mediator is released. EPA and DHA alter local packing characteristics, membrane flexibility, receptor clustering behavior, and the way proteins move within the bilayer. That matters because the inflammatory response is shaped from the start by how cells interpret their environment through membrane surfaces. A phospholipid environment containing more EPA and DHA is not reading stress through the same material conditions as one dominated by omega-6 derived substrate alone. The cell is receiving the same world through a different surface. Receptor behavior, enzyme access, membrane microdomain dynamics, and downstream signaling probabilities all shift. This means omega-3 incorporation into phospholipids influences both the early interpretive phase and the later mediator phase. It affects how the tissue enters inflammation and how well it may later leave it. This process is slow enough that it also explains why omega-3 biology is often misunderstood. People expect rapid effects from what is actually a remodeling event. Phospholipid incorporation requires digestion, absorption, transport, tissue delivery, membrane turnover, and selective replacement of existing fatty acid content. In a body long dominated by omega-6, heavy membrane composition, EPA and DHA do not immediately take command simply because intake increases. The phospholipid pool has to change over time. That means inflammatory potential is being remodeled gradually, not overridden instantly. The body is slowly replacing part of its future signaling reservoir. This is one reason the effects of omega-3 can feel delayed, partial, or dependent on broader context. The key question is not whether EPA and DHA enter the body once. The key question is whether they entered phospholipids deeply enough, broadly enough, and stably enough to alter tissue behavior in a durable way. Oxidative stability becomes crucial at this point because incorporation alone does not guarantee preserved function. EPA and DHA are highly unsaturated, and once placed into phospholipids, they contribute both resolution potential and chemical vulnerability. If the membrane environment is heavily oxidized, metabolically unstable, or repeatedly exposed to inflammatory damage, those incorporated fatty acids may be injured before they can support strong downstream conversion into pro-resolving mediators. This creates one of the central tensions in membrane biology. The same phospholipid enrichment that increases resolution potential also requires a tissue environment capable of protecting that enrichment from peroxidative loss. The body therefore needs more than omega-3 entry into membranes. It needs membranes stable enough to preserve omega-3 as usable future substrate rather than as easily damaged material. Seen clearly, EPA and DHA incorporation into phospholipids is the step that turns omega-3 from a dietary category into a determinant of inflammatory architecture. It places resolution substrate directly into the membrane reservoir from which future lipid mediators will be drawn. It changes receptor environment before activation and changes releasable substrate composition during activation. It is how diet becomes tissue and how tissue becomes future inflammatory behavior. Without incorporation, omega-3 remains mostly potential outside the structure. With incorporation, the membrane itself begins carrying a different answer to the question of what kind of inflammation this tissue will be able to run and whether it will be able to finish it well. Membrane reservoirs for SPM production. Specialized pro-resolving mediators are not produced out of empty chemistry. They are drawn from membrane reservoirs. This is one of the most important structural ideas in the entire resolution story because it means the body's ability to end inflammation is partly stored in advance inside its own phospholipids. EPA and DHA do not matter only because they circulate through the blood or appear on a supplement label. They matter because once incorporated into membranes, they become part of a tissue-level reserve that can later be mobilized when the inflammatory sequence reaches the point where completion must begin. The membrane is therefore not just the place where inflammatory signaling starts. It is also the place where the substrate for inflammatory ending is stored. A tissue can only generate meaningful SPM output if it has already built enough of the right precursor material into the right membrane pools before the need for that output becomes urgent. This gives the membrane a dual identity. It is a structural surface in the resting state, but it is also a biochemical inventory. When phospholipase activity is triggered during inflammation, the body does not release random fatty acids. It releases what the membrane has been loaded with. If that reservoir is rich in arachidonic acid and relatively poor in EPA and DHA, then the tissue is more prepared to generate early-phase eicosanoid chemistry than later-phase pro-resolving chemistry. If EPA and DHA are more meaningfully represented, the tissue carries stronger potential for downstream production of resolvins, protectins, and maresins once inflammatory sequencing begins to shift toward closure. That is why the concept of a membrane reservoir matters so much. It moves resolution biology out of the realm of abstract nutrient theory and into the actual storage architecture of the cell. These reservoirs are also tissue-specific. Different tissues build different phospholipid environments because they solve different structural and signaling problems. Neural membranes, retinal tissue, immune cells, endothelial surfaces, epithelial barriers, and mitochondrial membranes all handle lipids according to their own functional demands. That means the reservoir for SPM production is not identical everywhere. Some tissues may hold more DHA because of specialized membrane requirements, particularly where curvature behavior, excitability, and long-range structural stability matter. Other tissues may be more dynamically involved in inflammatory signaling and mediator generation. The body is not trying to make every membrane composition uniform. It is creating tissue-specific stored capacities for future behavior. Resolution potential, therefore, depends not just on total omega-3 status, but on where those fatty acids are actually being held and in what phospholipid context they are being stored. This storage logic also explains why resolution cannot be improvised well in a body built under chronic omega-6 dominance. If the membrane reservoir has already been filled disproportionately with initiation-heavy substrate, then the tissue enters inflammatory stress, carrying a stronger bias toward escalation. Even if omega-3 is later introduced, the reservoir may remain slow to change unless phospholipid remodeling becomes substantial enough to alter the releasable pool in a meaningful way. The body resolves from what it stored, not from what it wished it had stored. That is why membrane remodeling takes on such importance in chronic inflammatory states. Changing the future inflammatory outcome requires changing the reservoir from which that future outcome will be generated. The membrane reservoir concept also makes clear why oxidative stability matters so much. A reservoir is only useful if the contents remain intact. EPA and DHA stored in phospholipids increase resolution potential, but because they are highly unsaturated, they also increase chemical vulnerability if the surrounding environment is oxidatively unstable. A tissue under chronic redox stress may technically contain omega-3 derived substrate while still failing to preserve it as clean, usable precursor. Instead of serving as orderly reserve material for SPM production, those fatty acids may become damaged, fragmented, or converted into reactive byproducts that prolong inflammatory interpretation rather than support completion. The membrane reservoir must therefore be both adequately stocked and adequately protected. Resolution capacity depends on both conditions at once. Seen clearly, membrane reservoirs for SPM production are one of the deepest reasons membrane composition determines inflammatory destiny. The body carries part of its future recovery ability inside the phospholipids from which it is built. When inflammation begins, the tissue draws from what it has already stored. When resolution should begin, the tissue again draws from what it has already stored. If the reservoir is poor, oxidatively damaged, or crowded by competing substrate, completion weakens. If the reservoir is rich in preserved EPA and DHA and the tissue environment can support downstream conversion, the body gains access to the mediator families required to stop escalating and start finishing. The membrane is therefore not only a surface under stress. It is the reserve account from which the chemistry of closure must be paid. Structural prerequisites for resolution. Resolution depends on signals, but signals alone are never enough. The body can only complete inflammation if the tissue still possesses the structural conditions required to receive those signals, act on them, and survive the transition from defense into repair without collapsing back into renewed danger signaling. That is what is meant by structural prerequisites for resolution. Before specialized pro-resolving mediators can matter in a durable way, membranes have to remain intact enough to serve as stable signaling surfaces, phospholipid reservoirs have to contain usable precursor material, barriers have to retain or recover enough integrity to reduce repeated provocation, mitochondria have to maintain enough architectural coherence to fund cleanup and rebuilding, and the extracellular environment has to remain organized enough that repair is not immediately sabotaged by the residue of the inflammatory phase itself. Resolution is not just a biochemical command, it is a command that must enter a tissue still physically capable of obeying. The membrane is the first of these prerequisites because it is both the source of substrate and the surface of interpretation. A membrane that is too oxidized, too permeable, too compositionally unstable, or too heavily dominated by initiation-heavy substrate does not simply perform worse in a generic sense. It changes the direction of inflammatory behavior. Receptors interpret danger more easily and normalize danger more poorly. Phospholipids release a substrate pool biased toward escalation. EPA and DHA, even when present, may be too sparse or too poorly protected to support strong downstream conversion into pro-resolving mediators. The tissue therefore enters the later phase already handicapped. Resolution signals may still exist, but they are being delivered into a surface environment that remains structurally committed to the front half of inflammation. The first prerequisite for resolution is therefore a membrane capable of holding both stability and usable resolution substrate at the same time. Barrier integrity is the second major prerequisite because tissues cannot resolve while they are still being continuously reprovoked. An intestinal lining that remains too permeable keeps feeding microbial and luminal signals into immune surfaces that should be calming down. An endothelial barrier that remains too open keeps sustaining abnormal traffic, fluid shifts, and inflammatory exposure. Cellular membranes that remain damaged keep leaking distress chemistry into the surrounding environment. Resolution requires a reduction in incoming evidence that the emergency is ongoing. That reduction is not achieved through signaling alone. It requires boundaries that can close again. A tissue that cannot restore selective separation between compartments remains trapped in partial exposure, and partial exposure keeps the inflammatory program alive even when part of the later mediator environment has already begun to change. Mitochondrial integrity is another prerequisite that is often missed because resolution is too often described as though it were mostly about the right lipids or the right cytokines. Cleanup is metabolically expensive. Efferocytosis costs ADP. Membrane repair costs ADP. Restoring gradients, rebuilding junctions, processing damaged macromolecules, and reorganizing tissue behavior all cost ADP. A cell with structurally compromised mitochondria may still initiate inflammation because initiation can run on acute signaling, stored substrate release, and emergency physiology. But it will struggle to complete the event because completion demands sustained organized energy. Mitochondrial membranes therefore matter directly to resolution potential. If oxidative phosphorylation is poorly supported because the organelle itself has lost structural coherence, the tissue may receive pro-resolving signals and still fail to execute the work those signals are directing. There is also a mechanical prerequisite. The extracellular and local tissue environment has to remain rebuildable. Inflammatory events damage matrix structure, alter fluid dynamics, distort local signaling gradients, and generate debris that can physically interfere with restoration. If the matrix is heavily degraded, if tissue edema remains uncontrolled, if oxidized debris remains embedded in the site, or if repetitive mechanical stress keeps re-injuring the same environment before repair stabilizes, the body cannot move cleanly from inflammatory de-escalation into durable reconstruction. Resolution, therefore, depends not only on stopping further inflammatory recruitment, but on preserving enough structural order that restoration can take hold once recruitment slows. A tissue too thoroughly destabilized during the acute phase becomes difficult to resolve because the ground under repair keeps shifting. Macrophage competence also belongs in this structural category because effective cleanup is not only a signaling question. It depends on whether the cells tasked with finishing the event can physically engage, engulf, and process what has accumulated. If macrophages are metabolically strained, overwhelmed by debris burden, exposed to a persistently distorted mediator environment, or embedded in tissue whose architecture makes cleanup inefficient, then efferocytosis weakens even when some pro-resolving signals are present. The body needs more than instructions telling macrophages to finish. It needs macrophages structurally and energetically able to finish in a tissue that can actually be cleared. This is one reason chronic inflammatory states become so self-reinforcing. The site becomes progressively harder to clean, and that growing cleanup burden becomes one of the reasons the site keeps acting inflamed. Taken together, these conditions show why resolution is never just the second half of the signaling story. It is the second half of the structural story as well. The body can only exit inflammation if the tissue has preserved enough membrane integrity, enough barrier control, enough mitochondrial function, enough matrix order, and enough cleanup capacity to convert biochemical instruction into real biological completion. Without those prerequisites, even valid pro-resolving signals remain underexpressed. The body may try to stop escalating, but the site continues behaving as though escalation is still necessary because too much structural evidence of danger remains in place. That is the deeper meaning of resolution failure. It is not simply a lack of stop signals. It is a lack of tissue conditions that allow stop signals to work. Seen clearly, structural prerequisites for resolution explain why inflammation cannot be solved only at the level of mediator discussion. The membrane has to be built properly. The barriers have to close properly. The mitochondria have to function well enough to fund repair. The tissue has to remain cleanable and rebuildable. Only then can the chemistry of completion become the biology of completion. Resolution is therefore not just something the body wants to do. It is something the body must still be physically capable of doing. Lipid pools as signal libraries. Lipid pools function as signal libraries because the body stores future instructions inside its membranes long before those instructions are needed. This is one of the most important ways to understand inflammatory biology at a deeper level. A membrane is not only a boundary, and not only a structural surface. It is also a curated archive of possible responses. The fatty acids esterified into phospholipids determine which kinds of lipid mediators can later be released, which enzymatic pathways will have meaningful substrate available, and what kinds of downstream tissue behavior will become easiest to generate once the cell is activated. In that sense, the body does not invent inflammatory language from scratch every time it encounters stress. It consults the lipid library it has already built into tissue. The quality of that library determines whether the cell is better equipped to escalate, better equipped to complete, or trapped in a chemically biased middle where escalation is always easier than resolution. The idea of a signal library matters because it shifts the focus from momentary triggers to stored potential. Arachidonic acid-rich membrane pools represent one kind of library. They are heavily stocked with the substrate needed for eicosanoid production, vascular change, neutrophil recruitment, pain sensitization, and early inflammatory amplification. EPA and DHA-enriched membrane pools represent another kind of library. They still permit initiation because the body still requires initiation, but they also provide a deeper archive for resolvins, protectins, maresins, and the wider chemistry of inflammatory completion. These are not separate systems floating beside one another. They are different volumes on the same shelf. When phospholipids are activated, the body begins pulling from a collection it stored. If the library is dominated by activation-heavy substrate, then inflammatory meaning will be drawn mostly from activation-heavy pages. If the library is richer in omega-3 derived precursor pools and sufficiently protected from oxidative loss, then the body has a stronger basis for consulting the language of closure once the early phase has done enough. This library metaphor also clarifies why membrane remodeling changes future behavior even before any obvious symptom shifts. The body is always editing its archive. Diet, oxidative stress, phospholipid turnover, lipoprotein transport, mitochondrial strain, hormonal state, and repeated inflammatory events all change what is being stocked, what is being lost, and what remains easy to retrieve later. A tissue repeatedly built from omega-6 dominant input and repeatedly exposed to inflammatory provocation will gradually accumulate a library more fluent in early-phase inflammatory grammar. A tissue that incorporates and preserves meaningful EPA and DHA in its phospholipid pools builds a broader library, one that includes more of the language needed for mediator class switching, macrophage cleanup, tissue protection, and resolution. This is why chronic inflammatory states often feel chemically repetitive. The body is drawing from the same narrow shelf over and over again because that is the shelf it has been stocked to use. The phrase signal library also makes clear that stored lipids do not merely determine what can be released. They influence how signals are interpreted before release occurs. Membrane composition affects receptor behavior, microdomain organization, enzyme access, and the physical conditions through which extracellular information becomes intracellular action. That means the library is not only a future mediator archive, it is also a present interpretive framework. The surface of the membrane is already shaping what kinds of signals appear meaningful, how strongly they are amplified, and how easily the cell crosses the threshold into inflammatory activation. A library rich in unstable, initiation-heavy substrate does not only produce certain mediators later. It can also create a present-tense bias toward overreading danger and underreading completion. The cell becomes both more fluent in escalation and less fluent in restraint. This is why oxidative damage is so destructive to the library concept. A library only helps if its contents remain intact and legible. When PUFAs are oxidized, the stored archive begins to degrade. Clean substrate for controlled mediator generation is replaced by reactive fragments, distorted lipid signals, and membrane instability. Instead of preserving useful instructions for later resolution, the tissue begins carrying damaged text that keeps generating biochemical noise. The body can no longer consult its stored omega-3 derived potential as effectively because part of that potential has been chemically shredded. In that state, the tissue does not just lose signal options. It begins accumulating false signals as well because oxidized lipids themselves can act as evidence of ongoing damage and help perpetuate inflammatory interpretation. Seeing clearly lipid pools as signal libraries is one of the best ways to understand why membrane composition determines inflammatory destiny. The cell responds not only to what happens now, but to what has been stored for later use. Its membranes contain a history of previous dietary patterns, previous remodeling, previous oxidative burden, and previous inflammatory sequencing. That history becomes available the moment the tissue is activated. If the archive is rich in preserved EPA and DHA, resolution potential rises. If the archive is dominated by omega-6, heavy initiation material, and damaged by oxidation, the body becomes more likely to keep repeating the first half of inflammation. The tissue does not merely react. It reads from its stored library, and what has been written into that library determines how well it can move from alarm into repair without getting trapped in alarm itself.